Hi everybody. Today there is a new session in this YouTube channel about medicine of the future or personalized medicine. Today will be a bit different from the last two sessions. We were talking about personalized medicine. We were focusing mainly on cancer and AI, how it will make the future of medicine very different from now, right? So today we were talking about something different. We were talking about a paper, which is also related somehow to personalized medicine. We will be focusing on CRISPR. However, this is a bit different because this is one paper which shows a development of tool. This tool actually, it mix a bit between, this is a mixture between a CRISPR-Cas or CRISPR gene editing and GPT or ChatGPT. So we will discuss a bit about our invite today, our guest, Dr. Mohammed Ahmad Yamil. I will let him to introduce himself and then we can go further. Go ahead.
Hi Lubna and hi everyone. I'm Dr. Mohammed Ahmad Jamil. And thank you so much for inviting me to your podcast. It's truly an honor to be here. I am a bioinformatician and have completed my PhD from University of Bonn. And I know Dr. Lubna through our professional work, and I'm really looking forward to share my insights with you today about this very nice paper.
Thanks too much for this introduction. Yes, and let's go with our paper. So our paper is about CRISPR-GPT for agentic automation of gene editing experiments. So this paper was published in 2025, so quite recently, I think, in, I don't know, probably in July. In a very well journal, Nature Biomedical Engineering, where mainly they combine the workflow, or they make a workflow about CRISPR and GPT or ChatGPT. We will talk about that some more a bit in detail, but I would like to give an introduction about CRISPR. So what is CRISPR? Mainly CRISPR is a powerful gene editing technology. I think everybody knows what is CRISPR, a gene editing technology which was discovered some years ago from Doudna and the Emmanuel Charpentier in 2020. And this is basically the immune system in bacteria. It's mainly the bacteria use that for self-defense against viruses. And that's the strategy mainly. It's how to use this strategy, how the bacteria is doing in editing anything in the genome. So mainly the CRISPR-Cas is a gene editing system, as I say. It's used an RNA guide. So mainly there is one guide RNA which shows exactly where to target in the genome. And mainly this is how we can modify the DNA, and it allows the scientists to cut, delete, and insert or replace specific DNA. As I said, this is a revolution in medicine. This is also part of the personalized medicine, right? Because this is what we use to target a specific part in the genome, whether the gene or a regulator or any part we would like. Also RNA, and also we can do now epigenome. So we can introduce some modification, and this mainly it's how it works. So it works with, as I said, with the guide RNA, which match exactly the target of DNA we want. We can also target RNA, as I said. It has a different enzyme. So here I just cite two enzymes, which are Cas9 and Cas12. There are also other enzymes, and there is also Cas for epigenetic. And as I said, it makes a different cut or modification of the target, and mainly the enzymatic repair of the DNA, which allows this introduction into the genome. Why or how do we use, or what are the applications in CRISPR? So I think the first thing we all know, this is mainly in medicine, right? So how we can cure diseases, how we can make some correct genetic fingerprint, for example, fingerprint somewhere in the genome to avoid any mistake. And then we use it also for improving crops. So in agriculture, this is a very important point, and I have myself worked also in something which is related to crops and how we can do the bioagent control. For instance, we can edit any genome related to some fungi or insect or worms which are attacking crops, and then we can also edit some organism to attack the one who is attacking our crops. We can also engineer our crops themselves to make them to have the self-defense. So there are many strategies we can apply mainly in agriculture. Again, developing gene therapy and cancer treatment. Again, this is all related how we can avoid, for instance, some diseases, or we can repair or correct some variant. So mainly it's we can also create some disease models, right? So disease models in the research is very important. We need some model in the research to make them as a template and how we can find a cure. So mainly if we can create diseases model, we can easily also not only find the cure for these diseases, but also get a hint. It gives a hint about other diseases. And obviously exploring the synthetic biology and microbial engineering. There is nothing which is completely, I would say, without no limitation, limitation free. Obviously, we have always some consequences, and mainly we have here the consequence like off target. And here I think we have an expert with us which mainly work with these off target effects. What are the off targets? The off target mainly they are. We designed the experiment for on target. On target, this is mainly what we need to target, right? So we need to target RNA. So we designed for this part on the genome, regulatory or gene, whatever. But then we have off target. That means there are some part in the genome which could be affected because of the homology or because of the experiment, because of the technique, etc. So all these kind of parameters could lead to off target. But sometimes the off target could be also a hint about a new disease because there is a discovery about something different. It was not and purpose, right? So the off target sometimes open the eyes, open our or reveal new discovery, because this is somewhere in the genome which we didn't know what is the function, right? And now we know it because of off target. So of course there is a limitation because we want some on target, but also off target could be advantage in some time. We have also limitation of delivery, right? So the CRISPR component into the cell, how we can bring how we can get the CRISPR component into the cell an effective way. So this is also a complex topic. We have immune system, we have many barriers in our body, right? So that makes also some attack from immune system, also how the substances that one find in our system, which could lead that this CRISPR component will not reach exactly the part on the genome we needed. Then there is ethical concern. Of course, this is I think this is one of the major concern in human. So especially the human germline editing, how we can edit a fetus, for instance, because of the diseases, because of the ethical. Also, we want some babies, design some babies with blue eyes, or a baby more intelligent than others. So we can manipulate the gene, obviously. Although we don't know all the genes, we know we have only 20,000 genes for all the proteins we have, or all functions we have. We don't know everything, obviously. So attacking or targeting something could lead to something else. We don't know the regulators in our genome. This is one of very fundamental issues in our body, right? So the fact that we have only 20,000 genes and we have an enormous number of proteins, it's a questionable, right? That means there is only regulation effect or transcriptomic effect and post-transcriptomic atomic effect, which lead to all these broader functions we have in our body, right? So this is a really problematic. That means we can target some gene which we don't know afterward how the effects, right? So we can shut down a disease, we can switch on another disease which was not known before. So this is one of the, I would say, most of the ethical issues we have when we touch the genome, but obviously also the ethical is preferences, right? Why should be a baby better than others, or why should we create humans better than others? There is then society problems there because of we will create different classes, right? We have already different classes in society, but adding on that genetic background is quite complex. Yeah, so we have also the precision. Some edits are still difficult or inefficient, obviously, and the long-term safety and regulatory approval. This is also another issue, because we don't know how much is that stable in our body. This is one, and secondary, this is regulatory approval. That means once we have the treatment, we know all the FDA and the preclinic and clinics and so on. All it takes a lot of time. Then this is also something which I would say make some handicap. This is basically the paper we would like to talk to today. This is about the overview of CRISPR GPT. As I said before, the workflow it's about automation of the entire CRISPR gene editing process using the AI agent. So mainly these people, what did they do? They try to make a workflow step by step for the user, how to use the CRISPR combining with the large language model. So the large language model, in this case, they use the ChatGPT. So mainly the user has access to I would say the experiment. Before doing the experiment, has already all the steps written, how to make this experiment, what's the issue he can face, then he can talk or directly chat with ChatGPT, and he can handle mainly each step with the precise, with accuracy and so on. This is mainly how this paper is talking about. So obviously there are different, I would say, different limitations, and this is what Dr. Ahmed will talk about later. But mainly, as I say, they did effective test automation. So this is a workflow step by step, where they show the system selection, the enzyme, the guide RNA, the protocol generation and analysis, and they combined this large language model planner with the task executor, with the proxy, with the tool provider for complex reasoning, and then they try to validate their method. They use 4 knockout genes, and they use also activation of 2 genes epigenetically, and also they compare the ChatGPT 4.0 and ChatGPT 3.5, and they show that there are some improvement with ChatGPT, of course, with the 4, because it's more performant. But then they have, as I say, they have also some limitation. And I think these limitations are very, very important to talk about. Why? Because simply there are many research actually right now where they use this, I would say, they use these fancy titles, right, Ahmad, because these AI and LLM, large language model, and so this is very fancy for the people who did not deal with it. Exactly. So using this fancy title does not give a credit that this tool is wonderful, right? Does not give a credit that this tool will work forever. So I would not go really into detail about this workflow and so on. I think this is the measure idea, the main idea, that they combine both to give access to people, technician, biologist, expert, to how they can use this tool. This tool has also, as I said, different steps where the user can go and can always chat and can have some advices if there is if there is an error, could be probably corrected because the ChatGPT would propose you, and so on. And all of that it's done in a large data. This data was curated, obviously. This data was also under supervision of experts, as they mentioned. And so there are many limitations. I would leave now the mic to Dr. Ahmad. He can probably talk about what could be the limitation of these kind of tools. So how do you see that as an expert in CRISPR-2 and off-target, on-target, and so on? How do you see the consequences of the limitation of such tools?
First, thanks a lot, Lubna. It was a really nice intro and a nice overview about this publication, CRISPR GPT. So the first thing came in my mind is whether we can call it a tool or it's a guide. Because if we talk about a tool, we can say, okay, for example, CRISPResso is a well-known tool where you can analyze this. But I'm not sure when we talk about CRISPR GPT, is it a tool or it's a guide? Because when you were showing the overview, I can see that they mentioned some tools which they are using. For example, they mentioned primer thief using this to identify the off targets. So I think calling it as a tool is not really the right or correct nomenclature, but we can rather call it as a guide. And while we are discussing it, I also looked into this AI agent on CRISPR on ChatGPT. And I saw that when you start this thing, it will start asking you some basic questions. So it will guide you throughout the process. I think this is nice for the people who are really naive and who don't know much about the CRISPR and how you can go through. And I think also you mentioned in your talk that when they do the benchmarking, not against other GPT versions, but when they test it on some junior researchers, they found that, okay, this is working fine and they get the results what they desire. But still, I'm thinking how better it can produce for someone who has really basic knowledge of CRISPR. For example, I have been working on CRISPR for so long, so I'm analyzing on targets, off targets. So if we discuss, like, CRISPR usually used for two main purposes. One is diagnostic, another is therapeutic. So I worked for both parts. So we proposed some therapies, and we also did some analysis about the diagnostic, like, okay, if we have off targets, how it can react. But I'm thinking how it can help for a researcher or for a new PhD student. If he wants to go into CRISPR, he has some idea how it can help it. So maybe you can also comment about it, like if we thought about a new PhD student who has no idea about CRISPR, how this AI agent will help that particular student.
Yeah, indeed. This is one of the main points, actually, and thanks for this resume. Actually, it is one of the concern. I mean, who is willing to work with this kind of workflow, right? Are they experts? Are they really understanding what is the CRISPR? What is the impact if there is any error? And what is the impact also? What is error not only from the technician or PhD? What is the impact of error of ChatGPT? We know this is a large language model, right?
This is nice that you are thinking that who is the target audience of CRISPR GPT? Are these junior researchers or are these experts? Because if we talk about experts, they have the idea in their mind that how we can go through the pipeline or how we can go through. But what about junior researchers? Because they don't have. So you also think, because I think now that the audience for this CRISPR GPT is not the experts, but very new people, or the one who has no idea about CRISPR.
Indeed. And I think this is also an issue, because if they have no idea about CRISPR, obviously anyone can go to the lab and follow some recipe, right? This is like a kitchen. But then, if something happened on this kitchen, I mean, we can make eggs and we can fail and we can have something which is eatable or not. But CRISPR here, we're talking about experiment, could be really lead to some issues which will be published later, and nobody will be able to detect where the issue is coming from. And also something what I see, for instance, in these kind of pipelines, who is willing to maintain this pipeline with the huge statistic and dynamic field of biology? Biology evolves super rapidly, and also the how we would introduce that, I mean, constantly in this pipeline, because this is introducing each data which is created, which from under supervision of experts and so on. It's not easy task. We're talking about training the data from the beginning. Before training the data, we know as a computational biologist, we have to pre-process the data, which is also another headache. And also not only headache, this is something which is which is error prone, right? So who is willing to do that? It's also financially a problem. I mean, especially if it is kind of tools they are developed by university, academia. We know people are changing constantly. Who is willing to deal with this kind of a huge workflow which need time? I mean, they are costly, timely, and expensive also financially. They need expert to maintain it. So it's not that just like that, and because you have an AI or GPT in your title, that means this workflow will work fantastic forever.
Maybe they can add a module, because I think you can also use ChatGPT as a search engine. And I also, in their CRISPR GPT agent, they mentioned that you can find the latest research on CRISPR right now. So maybe they can add a module here which can do a search engine, just like normal search engine. They find the new paper related to CRISPR, and then they automatically, because this is LLM, of course, we can, using the LLM, we can do the machine learning from the paper. We can extract the information from the paper, and they can, rather than putting some guy or some expert there, they can also make an AI in that way. This is one possibility, but of course this is going into the next step of this particular tool.
Yeah,
again,
indeed.
I'm calling it tool.
Yeah, tool. Well, it's I would say this is like Lego, right? This is combining something with something and providing playing a Lego, but the Lego themselves, they are some blocks, right? Then each block is coming from somewhere which was developed by some group and so on. Yeah, another thing which I see here in these papers, so there is only fork knockout and epigenetic activation of 2 genes. This is really a very
short test set.
Very short and to validate really a workflow, which is a huge workflow like that, to provide to people to work. I mean, we cannot really validate something with just 6 experiments. I mean, this should be done in different way, different genes and different techniques, because also they use a prime 3, I remember. I think it was already mentioned here. This is let me check it. Yeah, this is I think they use here prime 3 and another tool which they use, for the editing. What about the future? I mean, I'm talking about the future some years, because each time we have a different tools. If the tool will be also behavior, I mean, the outcome of the tool will be the same, that it will be treated by the workflow and we have the same output. So there is a lot of discussion here, which or limitation too, that's lead really to think about such kind of workflow and such kind of publications. They come like bomby, because they are AI, LLM. People are not really aware that everything is coming at once, but we should really be careful who is willing to use this kind of workflow, who is responsible for the errors, actually.
I'm really reluctant about the thought that if an expert is using this thing, then it's okay. because he can later criticize the output. He can interpret the output what he get from CRISPR GPT. But as we are thinking that this is the target audience is not an expert, but the target audience are the naive people or the people who has no idea about CRISPR.
Yeah.
In that case, they cannot really identify false positives here, which is a high possibility to get one. Because if we thought about off targets analysis, this is off targets are all which could be positive, or it could be just an artifact of the sequencing or some other errors which can happen in the lab or in the analysis. So how a new person who has no idea can interpret such results? And for example, if we get 1,000 papers out of it and all of them has these problems, then what we get in the end is not really reliable.
Absolutely. I mean, there are many criticism here. Again, the large language model is still, this is a deep learning, right? So we're still improving the algorithm, but what about the hallucination? For me, it's still really something which is critical, because we know all the AI, how it works, and we know the algorithm, how they are constantly improved. But still, they are algorithms, right? And still, there will be always some error. And I think we all know from ChatGPT, if you can drive ChatGPT sometimes in your discussions, right? You can say something and he can give some answer, and later on he can say, oh no, I was wrong. I was saying, and indeed what you are giving is the wrong way, just to mislead him. And in the end, it will go with your, logical, and we say, yeah, you're right. What about if it happens here? I mean, this is really,
so
this is really critical.
Here, think about two more things. One is ChatGPT is highly based on the memory. So whatever you are giving it as an input, it stores it and it gives output according to that. And the next thing what we have to really worried about is the prompt which we are giving to ChatGPT. So as you also mentioned that we can mislead ChatGPT directly, of course this is high possibility. Then even for the right answer you can say, okay, Mr. blah blah, you are wrong and this should be like this, and ChatGPT will agree with you, because I think it's in their nature how they design it, that it agrees with the asker, like what he is asking. So I think it depends on the prompt and the memory, how it is designed, and based on that, I'm not sure the result which we get. If it's keep getting trained on false data or incorrect data, then I think in the end we will just get garbage out of it and nothing really useful.
Absolutely, this is another issue, right? So the data which will be then processed later, it should be, with a high quality and also filtered and so on, that it could be again trained to have a better outcome. Otherwise, we will just accumulate in garbage, as you say. The garbage is the proper word. I thank you for that. But this is the risk of such a workflow. Obviously, as you say, who is willing to work with that? Who is the consumer?
Who is the consumer? Yeah.
Who is the consumer? Is it the PhD? Is it experienced, not experienced? Because we will not expect some experienced using that, right? I mean, what is his experience? It is elaborated, or it is developed for people who are not that experienced. So here it is a dilemma, right? What is what and what for?
In their publication, they mentioned that they designed this and they get the output, and it was perfect. With high efficiency, they get the output. So I think this is nice tool for designing the experiment, but you need a supervision here. I think in that way we can be safe to say that, okay, a person can use this tool if he or she has a supervision.
Exactly. Exactly. I would totally agree. So probably they have to mention that in the conclusions, because this is of the main point. Don't use that without supervision, because you may lead to some experiment which is useless, or in the worst cases, which is harmful. I mean, useless is something, because also it's money and time, but harmful, it's another level. So yeah, I think we covered mainly the most important topic about how could this workflow be successful and at the same time harmful, right? And just to make the idea to the audience, don't be misled with just AI, GPT, LLM, and so on. We need really to understand what is the background and what is for. So using something just because, or using now this kind of fancy word and research, it's a bit, for me at least, it's a bit crunchy. It's a bit crunchy, because it's not really what is the aim. I mean, again, in science, we have been using that a long time ago. I mean, it's AI is not something which is I mean, I think all what we are using in variant calling, for instance, as we are doing, or anything of VGS, RNA transcriptomic, and so on, it uses AI to develop these tools.
My experience from my PhD. So for me, AI was an intelligent guy. So for example, my supervisor. So if I have some question, I go to him. So for me, AI earlier was an intelligent guy. So he was helping me all the time. It's same like here, we are using AI. But the problem between this AI and the AI in the past was, the AI in the past was experienced, he has knowledge. But what AI we have right now, it's based on the knowledge we gave it to him.
Exactly.
It has some limitations.
Exactly, absolutely. So the conclusion, just be aware, there are some fancy work done in research. Obviously, the AI will have been used, or has been used for a long time ago, probably late of 80s or beginning of 90s, at least in medicine. But now how the research is using AI as a title to attract people and so on, it sometimes lead to question. So how would you rate, Ahmad, the paper?
So I like the idea to introduce AI, and of course it's helpful rather than searching on Google how we can design. It's good. But as we can see the limitations and the ethical problems in all this, and the fact that it can produce some false results, so I'm not really happy with their conclusion that they should mention about this, like the target audience, who should be target audience, and who they should be supervised and things like that. And they need curation, they need someone always on it who can update it regularly. So based on the new technology, based on using AI and things like that, so I would suggest to give it 6 out of 10, I think.
Okay, I wouldn't give it more than that. I think we agree about the about the 6. The 6 is not bad, but neither I wouldn't give it more. I think it's fine. Okay, so we will reach our end. Thanks a lot for this rich discussion. We will talk about many papers more. So thanks a lot. Thanks for the audience, and we'll see each other next time. Thank you. Bye bye.
Thank you. Bye.